While vaccines are one of the most powerful public health tools for protecting against infectious disease, not everybody is conferred the same level of protection. Many factors determine whether an individual responding to vaccination will generate an effective response, including specific biomarkers within a person's immune system, but until now there has been no evidence showing whether these factors were universal across a wide range of vaccines. New findings from a meta-analysis published in Nature Immunology examine the biological mechanisms responsible for why some people's immune systems respond differently to vaccinations, which could have global implications for the development and administration of vaccines. As part of a series of studies for the Human Immunology Project Consortium, HIPC, a network of national research institutions studying the range of responses to different infections and vaccinations, Emory researchers analyzed the molecular characteristics of 820 healthy young adults who were immune. The participants were separated into 3 endotypes, or groups with a common gene expression, based on the level of inflammatory response prior to vaccination: a high inflammatory group, a low inflammatory group, and a mid-inflammatory group. After studying the immunological changes that occurred in participants following vaccination, researchers found the group that had the highest levels of inflammation prior to vaccine had the strongest antibody response. "We were surprised because inflammation is usually depicted as something that is bad," says Slim Forati, Ph.D., Bioinformatic Research Associate at Emory University and first author on the paper. These data indicate that some types of inflammation can actually foster a stronger response from a vaccine. Forati, Dr. Rafik Pierre Sekali, professor and senior author of the paper, and the HIPC team identified specific biomarkers among this group and cellular features that characterized the P. With the knowledge we now have about what characteristics of the immune system enable a more robust response, vaccines can be tailored to induce this response and maximize their effectiveness, says Forati. But we still have more questions to answer. More research is needed to determine the cause of this inflammation in otherwise healthy adults. Additionally, Forati suggests future studies should look at how these biomarkers facilitate vaccine protection in older age groups and among populations who are immunocompromised. Published simultaneously with 3 other HIPC studies by researchers at Yale School of Medicine, Stanford University, University of Cincinnati, Harvard Medical School, and Columbia University Medical Center, these findings can serve to improve vaccine response across all individuals. Better understanding of how various pre-vaccine immune states impact antibody responses opens the possibility of altering these states in more vulnerable individuals. For example, scientists may give patients predicted to have a weaker immune response an adjuvant with the vaccine to trigger the inflammatory genes associated with greater protection. This work will help enable improved, more efficient clinical trials for the development of new vaccines. The HIPC program was established in 2010, renewed in 2015 and 2021, by the NIAID Division of Allergy, Immunology, and Transplantation. Thanks for watching. Please subscribe my channel for more videos.